All right, so we've taken the plates out of the incubator. Now we need to pick the colonies and let them grow overnight. So here I have 6 tubes. I've labeled 5 of them, so I'm just going to label this last tube. Okay, so now what I have is 6 tubes. I have 3 experimental colonies that I'm going to pick from the experimental plates. I have a positive control, a negative control, and then just LB amp, because I want to know what sort of signal I'll get from just the media. So I've labeled all the tubes, and now I have the plates. Here's 3 of them. Here's the experimental. Here's the positive control, the negative control. So I will start with one of the experimentals. And these are sterile pickers, and there's a loop at one end, but a fine point at the other end. So I want the fine point. I'm going to open this up, just grab it, and put this here. More slipping. All right, there we go. All right. So now what I'm going to do is I'm going to look for a red colony, and I'm going to just touch it to the tip of this plastic probe like that. So now the cells are on the end of this tip, and here's my experimental 1 tube. And so I'm just going to put the cell in there and then slosh it around. I just need some of the cells to come off this little picker. Put the cap back on, toss that, and move it. So now I've picked 1 experimental colony. I'll pick the other 2 experimentals, positive, negative, and then the LB. I won't add anything. And I'll put it in the incubator and let it grow overnight, and we'll see what we get.